I want to compliment Nuno Martins for organizing this incredible summit. For those who don't know me, I founded a group called Life Extension back in 1977, and we have a track record of developing and identifying biomedical advances years and sometimes decades before they're recognized by conventional medicine. For those who want to see these slides I'm going to present, they're all available at age-reversal.net. That's available anytime to review the slides, maybe even see some live presentations I've given before on these topics. What I want to start off talking about is effective altruism as described in Forbes magazine back in 2019. And that is putting money into initial public offerings and where the money goes into real research as opposed to just spending more money on promoting the company. In this case, this brilliant scientist, Alex Zalvaranikov, was able to develop an AI technology to enable very rapid development of drugs, in some cases longevity drugs, to extend healthy human lifespan. Now, in silico medicine started around 2014 with Alex Zavarnikov's own money, and he pioneered AI, maybe for the first time, to develop drugs that could extend healthy human lifespan. But in 2016, he was running low on money, and he came to some charitable groups like we are, and we were able to contribute about $140,000 towards some research to advance his technology, advance the entire science. In today's dollars, that may equate to around $250,000. But for that rather modest amount of money, he was able to show that he could create a new molecule from scratch in just 21 days and validate it 24 days later. Absolutely amazing. This is the first time that the potential for AI to be used in the pharma industry was validated to move theory into practical reality. So what happened in exchange essentially for that incredible study that was published, he was able to raise $37 million of additional funding. And that then propelled him into a new league where he's partnered with Eli Lilly to secure nearly $3 billion in licensing fees to develop longevity drugs and other drugs to combat biological aging. So to review this chronology, 2014, Insilico started with some initial money, and some of their objectives were literally to find a cure for biological aging. They needed some additional funding, and nonprofit groups invested and donated some money into Insilico, and that motivated, after the successful research, venture capitalists putting the $37 million into Insilico. In 2026, Eli Lilly signs a multi-billion-dollar deal, and the market cap for Insilico Medicine is over $4 billion. And there are other companies like this that we've identified over the years, and some of them have done very well, others haven't. But in every case, we were able to advance the science to the next level to potentially enable us to live a lot longer. And to put this into numerical terms, $10,000 invested in Insilico in January 2017, and that money all went into research. Well, that grew to $297,000 by 2026. That's a 29-fold improvement in investments. And even more important, it advanced the science, which is what we're seeking to do. And what's so fascinating, Eli Lilly, one of the world's largest pharmaceutical companies, is investing heavily into longevity medicine. They want to develop anti-aging therapies, not only just to treat age-related disease, but to achieve healthy longevity for the whole world if they can, with the technology they're developed with AI technology and silico technology, assisting Lilly in developing brand new drugs. Now, the New York Times historically has been rather hostile towards alternative medicine and aging research. Well, they're changing their mind a little bit. They're talking about the rich and powerful wanting to live forever, which we think is a great thing, because if they get to live forever, they're going to sell that technology to us. And the wealthiest people in the world putting big money into aging research. New York Times reporting on a potential new therapy to cure diseases and even reverse aging. They're terming this cellular rejuvenation. It's transformative, and it may extend life by decades. This is New York Times being quoted. Fend off aging entirely. And that new therapy that they're talking about is something that they refer to as rejuvenation, one of the newest, most promising developments in longevity research. Well, we've known about this for a heck of a long time, and we're seeing it now recognized by people who in the past were rather cynical to what we have been seeking to accomplish. And the world's most successful, influential people are putting big money into this. Now, cellular rejuvenation, according to the New York Times, is what we term cellular reprogramming. And that's taking that old degenerated cell to the left and rejuvenating it back into a healthy young cell via cellular reprogramming, in our case with our research using the OSK Yamanaka factors. And Washington Post last year reported on the proper nomenclature. Cellular reprogramming is the method to partially reverse old cells back into a more youthful state. Shinya Yamanaka discovered this back in 2006. These Yamanaka factors, they reprogram old cells back into young pluripotent stem cells that can regenerate, theoretically, our tissues forever. And these Yamanaka transcription factors, cells were shown to go in reverse in 2006, live mice 2022, 2024. Monkeys, well, we started the OSK infusions into monkeys, and we're going to know results very, very soon. If those old monkeys grow biologically younger, we anticipate clinical projects being initiated in 2027 to take old people to reverse their aging the way that was done in the mice. The original Yamanaka factors, OSKM. We are using now just OSK. We think it's safer and potentially more effective than OSKM in inducing systemic biological age reversal. Now, the Salk scientists back in 2022, they used all four, OSKM, and they were able to take middle-aged and elderly mice and reverse their aging process, made international news headlines. For the first time, aging goes in reverse systemically in live animals. And then a couple of years later, another group used an OSK delivery system with old mice equal to about 77-year-old people, and they were able to reprogram their cells in a way to increase remaining lifespan over 100%, improve frailty scores, and other measures of aging. The control group in this study lived about 8.86 weeks, and then the ones getting the OSK treatment, their remaining lifespan, 18.5 weeks. It's about 100% improvement in remaining lifespan. Now, the in vivo delivery methods being studied, there's the viral vectors, infection, transfection of the OSK into the body. We aren't using viral vectors. We are studying polymer and plasmid-based vectors, lipid nanoparticles, chemical reprogram being tested by some other groups to see if they can endogenously induce the OSK expression in living people, living animals. Now, our research identified in mice the most effective way of transfecting. That means delivering OSK into nuclear DNA, the polymer nanoparticle, and also the lipid nanoparticle. These are able to encase the OSK blueprint in a way that they can be delivered to the nuclear DNA so that we can reprogram those cells back to youth. And this is a diagram that explains how we enable this to happen. We encase the blueprint for OSK into a lipid nanoparticle. And lipids, of course, they go through the outer cell membrane very easily, sometimes more easily than we want. But nonetheless, lipid nanoparticles, they go through the outer cell membrane. Inside the cell, they release the blueprint for OSK inside the cell. And that mRNA blueprint for OSK is transformed into OSK genes by the ribosomes that then go through the pores of the nucleus and bind to nuclear DNA, where they reprogram that old DNA to tell the whole cell, including the DNA structure, to function and behave in a younger state, rejuvenate the older cell via the reprogramming of the cell via the induction of OSK delivery or transfection into nuclear DNA. Now, our research was able to achieve some unprecedented results that will be published. We were able to show that the OCT4, that's the first of the OSK factors, that was able to induce immediate expression of the OSK, immediate delivery of that OSK into the body 6 hours later, 24 hours, 48 hours, 72 hours, and then we go days. We went up to 21 days, and we still had OCT4 expression in the liver of these live mice. And we had the SOX2 and the KLF4 also transfecting into the liver cells in a sustained way, a robust and sustained way. And we go into the heart. We were able to transfect the heart DNA with the OCT4 SOX2 KLF4. For people with chronic heart failure is a major cause of death, disability. We might be able to rejuvenate these old hearts back into a youthful function. Lungs were able to be successfully transfected with the OSK, and the spleen, OSK, systemic and sustained transfection. Skeletal muscle, wow, how much we need that with sarcopenia being a major problem with aging. If we were able to deliver the OCT4 SOX2 KLF4 in a robust and sustained method, it may way into the muscle. And chronic kidney failure, major problem. People with CKD, chronic kidney disease, that are hanging on, hoping not to go on dialysis. Well, we were able to transfect the kidneys systemically with the OSK regeneration factors, rejuvenation factors. We were able to achieve that. The big question is the brain. Were we going to be able to cross the blood-brain barrier and transfect brain cells with OSK? Fantastic news is we were able to do it using this nanoparticle delivery system. Doesn't involve a virus, doesn't involve chemical reprogramming. This is literally taking the OSK blueprint and enabling it to transfect neurons, brain cells, so we could potentially treat different types of dementia. Put those old cells back into a younger state. Can't wait to have it happen. We're studying it in monkeys right now. Live monkeys being given the OSK Yamanaka factors with both a polymer encased mRNA OSK or a lipid nanoparticle mRNA OSK. And we're going to have DNA methylation data relatively soon, along with immune function data, metabolic function data, and we want to see how long do these monkeys live. Our first injection was in December 2025. We've given them 2 more injections, 3 more injections, and our objective is systemic age reversal. We will have results relatively soon. We will disseminate that information to the world so they can see what we're doing, emulate it, improve it, do whatever we can to achieve super longevity. We're not the only group studying OSK. The multi-billionaires are, major universities are studying it. There is some indication there may be some human study this year using OSK in different ways to rejuvenate older humans. And I want to show this final graph here, this AI graphic. It depicts the old cell being rejuvenated slowly into a younger cell. And it does it highly accurate. Just one entry into an AI program was able to identify this type of graphic and display this type of graphic to motivate us to understand we want to take that old decrepit cell to the left and we want to slowly convert it to a young healthy cell, so we systemically grow biologically younger. And I want to thank the entire group for watching this little 12-minute broadcast of what is a lot of research our Life Extension group is funding. We're advocating that this research be supported across the board so that the whole human race can benefit. So thank you, Nuno, again for this incredible summit, and I look forward to further discussions.